nice to see a lot of people here still interested in peptides. There's a lot of oligo science happening, and it's nice to keep the momentum going for the peptides as well. So I'll talk today, continue the conversation on Fmoc amino acids. As you know, over the years, the transition has happened from Bach chemistry. I still did some Bach chemistry at the Weizmann Institute many years ago. And now we're all into the Fmoc amino acids. So we'll talk a little bit about control strategies, and also in the context of a manufacturing company, Polypeptide. We have multiple customers. We have a lot of commercial products. So how do we advance the science of controlling our starting materials, yet still consider supply chains that are active, commercial products, products in development? How do we do all this? So the next slide, please. So I'll talk first of all a little bit of a case study about a carbobenzoxy impurity we had in an Fmoc amino acid, and how it made its way through all the way into our crude product. And I think this will kind of emphasize why is it important to constantly update our control strategy and keep current, whether it's the modernization of techniques or the introduction of more high-resolution mass spectrometry that's now available, better separation science. This is all very, very relevant. And to the question about where do we start controlling things, we definitely have to start at the Fmoc amino acids because they make their way into our product. Then I'll talk a little bit about just general concept of control strategy for Fmoc amino acids, what we at Polypeptide have been doing to revise our specifications and update them. Specifications is just the more the end product. I mean, there's a lot of work that has to be done on analytical methods, and then specifications and justification of those specifications. So there's a lot of work that has to go beyond just developing the HPLC or HPLC-MS technique. We're a manufacturing company, and sometimes we have a plan, and then we have a customer that comes and asks for something, and we have to change our plans, whether accelerate it or change direction. So there's an interesting concept of we have our company that makes the peptides, we have regulatory expectations, we have our colleagues at USP, we have our suppliers. So I'll show you, it's kind of an interesting debate on how to move things forward. And then finally, I'll touch a little bit about how we implemented this new revised control strategy and some of the considerations we had to do. Let's go to the next slide. So quick case study. This is one of the amino acids that's pretty prevalent, aspartic acid, Fmoc, and it is routinely tested for, this is also by the supplier, but also now at Polypeptide. It's tested for traces of carbobenzoxy impurity, which is formed during the manufacture of the Fmoc amino acid. And as you know, TLC has been around for more than 100 years, and it's still a very good technique to recognize a lot of trace impurities. So TLC technique was implemented by the supplier to specifically control this impurity, and it's also able to detect free amines at a very low level, so it's still a very useful technique. We used this amino acid to make a small intermediate, which was then introduced into a larger peptide. And what we found out was that, unfortunately, there was a trace amount of this carbobenzoxy Fmoc amino acid building block that got incorporated and generated an impurity that by chance eluded exactly precisely at the retention time of our peptide intermediate. So when undetected, I have a little diagram at the bottom of the slide, we had a control strategy for the Fmoc amino acid, we had one for the peptide intermediate, and of course, our final API, we run UHPLC MS, high resolution MS. So in this situation, we actually had a control strategy not tight enough to be able to pick out this impurity and understand that by chance, again, a dipeptide, key peptide intermediate could form, and which the impurity exactly co-eluted, and it was just missed by the control strategy. And then we ended up detecting it in the UHPLC-MS of the final peptide. So this is an example of why we need to continue to develop and evolve our control strategy for Fmoc amino acids. And yes, our suppliers test for the purity and all the other attributes, but we conduct the testing as well. And we are the drivers of this control strategy because we are the ones who are working with customers, working with regulators, and going back to the suppliers and saying, well, we need to introduce new testing. We need to introduce new techniques. So it's an evolving a puzzle, so to speak. Let's go to the next slide. So here's a kind of, I tried to picture it as a diagram to help make this point. We as the analytical scientists, we can develop new methods, but it's in the context of multiple forces pulling at us. So we have, again, our customer needs and demands. We have what's possible to make and actually manufacture, and how suppliers can produce things for us at a certain purity level or certain specifications. Another interesting concept is we have an active supply chain. So imagine you're introducing a new HPLC method into testing at our sites, but the suppliers are using their own method, and we have to understand what they can do for us and which kind of techniques they can use. There's regulatory commitments because we have commercial products, so some of it's filed already, so we have to go back and convince regulators that our techniques are now superior. And so all this it's a big puzzle, and it's an interesting puzzle. So at the heart of the control strategy, yes, there's method development, impurity characterization, which has definitely become easier now with high resolution techniques. There's the understanding of what should the control limits be, and the understanding of the process and the justification and specification. As you saw, the guidelines coming out of USP, they're really helpful for us. This 1504, I had the opportunity to give some comments on that too. And in this context, how do we change and update? I guess the way I would describe it is you have a moving train and you have to change the wheels. So you have peptide manufacture that started, large-scale peptide manufacture that's been going on for the last 3 or 4 decades, and we have commercial products, generic products, and you have to change all this while you're still supplying the market, while patients are still needing to get these peptides. So it's an interesting puzzle. Let's go to the next slide. So some of the things that we were looking for, we wanted specifically a polypeptide to update our main control method using modernized UHPLC methods. And again, mass spec has played a key role here. Imagine all these impurities that you have, small trace impurities, and you're able to actually quantify them also by mass spec if you need to. So really powerful tools. Chiral separations, that's also improved in the past decades. So in the past even 5 years or 3 years, there's been tremendous progress in columns that are commercially available to perform chiral separation, which is fantastic. So there's a lot of new tools now available. Quantity, how do you quantify an amino acid? I know a lot of us think about solid phase synthesis in the context of, okay, we're using 3 equivalents or 4 equivalents, it's an excess, why is it that important? But in large scale production, when we're trying to look at the new incretins and new peptides that are coming out from pharmaceutical companies that are made in metric ton scale, just using 3 equivalents is kind of expensive. So we have to really be mindful about quantity, about how much are we using. We heard a lot of talks about environmental impact. So all these parameters and all these toolbox that's now available to us as analytical chemists become very important. And a lot of other methods to control parameters. So to the question that was asked before, yes, the Fmoc amino acids are key to control because everything ends up in the peptide. You see it all in the peptide. It's not missed. If you had a D-enantiomer, it'll translate through. If you had a foreign amino acid, it will go in. You'll get it in the peptide. So it becomes very relevant and important. Let's go to the next slide. So, analytical chemist. I had to give a couple of chromatograms, so it's okay. This is an older method that was used probably for the last 15 years to look at impurities in Fmoc-Asp OtBu. This is the same case study that I was presenting before. So we went ahead and modernized the technique by using a UHPLC column, so you get a faster separation, better resolution, and better resolution of key impurities, including what you see at around 8 minutes, just before the main peak. You actually see a OT-butyl, which is at the C-terminus and not at the side chain. So these little impurities that must be controlled. Now this is, of course, all spikes, so you're seeing here a small amount, or a spiked study where we added all the impurities into the Fmoc amino acid. So you really have modernized techniques that are able to elevate our ability to control these materials. And any of these impurities, if you don't have a good handle on them, they will end up in your drug substance. So it becomes really relevant and important. Let's go to the next slide. This is an example of a chiral HPLC. Now there's, again, modernized chiral columns that are commercially available. And in the context of a QC lab, by the way, the concept of a commercially available column is a little different than, I guess, in an academic setting, in that you want to make sure that the supplier of the column can actually produce that column multiple times, give you a consistent supply. And we many times want to have a backup supplier as well for our column, just because things do go wrong. And column chemistry is also a chemistry, right? Somebody has to build that column. It's got a stationary phase. It changes over time. So that's also a consideration. So here we had a separation that was a little bit borderline, that we could still do it, but we decided to modernize this technique. And also we wanted to eliminate, so this is some of the older solvents that still had hexane in them, and we just wanted to modernize it, again, for the safety of our employees. And we ended up with a newer method that's able to separate much better the D-enantiomer. And you can see at the back end of the chromatogram impurities that are not necessarily chiral. So a lot of the considerations are, you're applying a very, very detailed technique to look at a specific impurities, but of course all the other impurities that are there also have something to say. So they're also in this chromatogram. So there's two chromatograms just to illustrate some of the work that had to be done at the core, or at the center of what we were doing to modernize our control strategy. Let's go to the next slide. So here's an example of a specification, or part of it, it's a snapshot of the specification for Fmoc-Asp OtBu. And it's interesting that when you consider impurities, there could be impurities, and I'm talking about impurities that are related to the amino acid. So there could be impurities that are completely non-reactive, so they just don't react. And now it's a question, well, if they don't interfere and they don't react with the peptide, do I really need to control them at a very tight level? And that's where this justification of specification and risk assessment really comes into play, because you can envision how this impurity will end up in your final drug substance. Now, another point, just before we continue with this, is that a lot of peptide manufacturing companies, as ourselves, when we design a control strategy, Of course, we want it to be applicable to all the peptides we make, not only one peptide, because we produce multiple peptides, some of them commercial, some of them in development. So we'd like to have a unified control strategy, which is essentially the lowest common denominator. What you're seeing here is something that's also applicable to the generic peptides. So we had to control it at a very low level. And so you see some of these impurities are controlled now to 0.09% mole. And that's also a change that's happened, I think, over the years. A lot of times when you run HPLC, it's peak versus total, very natural. And in the context of a peptide, you wouldn't be wrong, because if you have a 30 or 40 mer peptide, if you have an impurity, so for example, deletion or a double addition, the molar extinction coefficient would not be that much different, and certainly well within the variability of your analytical method anyhow. But with impurities in the Fmoc amino acids, it could actually have quite a dramatic effect, especially if there's side chain protecting groups that are highly UV absorbing. So the conversion from percent mole to 2% mole, sorry, from percent area to percent mole is actually important. It's another step in the quality control lab. They have to do some calculations. Usually it's automated now with the new softwares. But everything now is changed to mole percent. So what you see here is a specifications for some of the key impurities, and some of them we could have decided to control to a lesser degree, but again, because of our generic portfolio, there was a decision to control everything down to 0.09. So interesting considerations. We have to change the control strategy. We have to have it evolve, but it has to be tailored to all the products that we make at Polypeptide. Let's go to the next slide. So we embarked on this project 2 years ago to revamp and rejuvenate our control strategy. And we were looking at the 20 Fmoc amino acids first, which are part of our global supply. As I mentioned, our suppliers do conduct testing, but we conduct all the testing ourselves as well, just to provide us additional assurance. And this was the first step of the process, really, to look at all the 20 amino acids. And we also looked in scope for 25 additional amino acids that are used by more than 1 or 2 sites. There's a lot of popular non-natural amino acids that are now being incorporated a lot into peptide pharmaceutical development. So the initial scope was about to develop 80 methods or so, and we were looking at a 36-month or a 3-year time frame to conduct this work. And our site in Malmo in Sweden was charged with this task. And as I showed you, we have all kinds of forces acting on us. So one of our customers came to us and said, well, we're actually manufacturing PPQ in 2023. So we want you guys to have it already. So, okay. So we ended up accelerating the project and conducting full ICH validations for all our methodologies last year and finishing it off this year. And we couldn't actually do all these validations in one lab, so that one lab could not take all this. So fortunately, we have multiple. We have 6 sites, so we divided up the work amongst the 3 European sites, and now we are implementing this control strategy. And this is another interesting part, is that we have a current control strategy. It's working. It's being used for commercial products. We have to implement this dual or second row of testing on top for this specific customer, but other customers are requesting it as well. So there will be a time frame of probably about 2 years where we'll have to run both methods and do some kind of bridging. These methods are far superior. The specifications are tighter. The justification of specification is more detailed. There's a lot of things that are improvements, but still we have registered products. We have commercial supply. We have to keep that going. So it's an interesting concept. And then my last slide, I think. Let's go. One more slide, please. And next slide, please. So again, just to finalize this point of the complexity of changing a control strategy while you're still using it, we had to continue our discussions with our suppliers. And as you imagine, when you're looking at 100 kilogram or 1 ton or something like that at scales, many times you will choose to have more than one supplier of an Fmoc amino acid. You don't want your whole supply chain based on one supplier. So this whole negotiation with our suppliers, what can be done? Here's our methods. They're more capable. Yes, you have to tighten specifications. You have to have better control of impurities. If you have all kinds of residuals, we need to remove those. So all these discussions are happening. So what I showed you, those 2 chromatograms are just kind of the center of the control strategy, but there's so much work to be done with respect to suppliers, customers, regulators, and keeping an active supply chain going. So with that, just a quick thank you. Next slide, please. I have a fantastic team of scientists working. Anna's leading it in Malmo, and another team in Strasbourg, and acknowledging the team in Brain that Didier is leading, and my colleague Jan, who's head of global development, and Landon Peluso, who's our quality assurance global director. And I thank you all for your attendance, and thank you.